Hello, everyone. My beautiful and handsome biologists. Teacher Sharuk, your favorite biology teacher, is back. And in this lesson, we are still covering cell structure. In this lesson, we are going to mainly concentrate on calculations. These calculations are very essential in your different papers that you'll be doing, paper 2, paper 1, even in paper 3, practicals. These are the calculations that you'll be brought. So if you're interested in getting that A star, getting the A, getting a really good grade in biology, stick around. You will not regret having stuck around. And in this section, we are mainly going to look at magnification, how to calculate magnification, how to calculate the actual size, how to calculate image size, how to use an eyepiece graphic to determine the size of an image. That is what we are going to look at. Don't forget the units that we are expected to use: millimeters, micrometers, nanometers. And I remember in our previous lesson, we looked at how to convert from one unit to another, from millimeters to micrometers, from micrometers to nanometers, or whichever way, depending on a question that has been posed to you. Remember that cells are really very small, and you can imagine that the smallest structure your eye can see is around 50 to 100 micrometers in diameter. And cells are way much smaller. Cells vary from 5 micrometers to 40 micrometers. That is the reason as to why you cannot see them with your naked eye. That is why you need a microscope to aid you in seeing these tiny, tiny structures. If a cell itself is small, how about the structures inside a cell? Like a mitochondrion. A mitochondrion is just 1 micrometer in diameter. And then there are even other smaller organelles which you cannot even see with a light microscope, such as the ribosomes. They are 25 nanometers. And you can imagine at the end of, you can see this full stop at the end of this sentence. In this full stop, you can line there over 20,000 ribosomes. So that is how small they really are. And we usually use a microscope to magnify and make these small objects become bigger so that our eyes can see them. So that takes us to what we call magnification. I'm sure you've heard about magnification. You've done some calculations from your IGCSE. Those who did not attend IGCSE still, we are going to be able to carry out these calculations very well. No need to worry that, oh, for me, I didn't attend IGCSE or what. No, I'm going to be able to take you through, and by the end of this lesson, you'll be able to understand. So when someone talks about magnification, we are looking at the number of times larger an image is than the real size of the object. Let me give you an example. You can look at yourself in a mirror. I'm sure some of you, or everyone almost, has been able to look at themselves in a magnifying mirror. Assuming you look at yourself in a magnifying mirror, you'll find that if the size of your head, your actual size, if this is your head, the magnifying mirror will make it much more bigger. So your head is the real size of the object, or your head is the object. Now, what you're seeing through this magnifying mirror is what we call an image. So this is an image, what you're seeing in the mirror. And you might be interested in finding out how many times has this mirror made my head bigger? So the number of times your head is made bigger in the mirror is what we call magnification. The number of times larger the head in the mirror is compared to your real head. That is what we call magnification. The number of times larger an image is than the real size of the object. Remember that microscopes carry out magnification. They make objects bigger compared to their real size. So we might be interested in finding out how many times has this microscope made this object bigger compared to its real size. And magnification can be calculated. So that takes us to the calculation. For you to calculate the magnification, it is very easy. It is very simple. As long as you know the size of the image and then the actual size, you can get magnification. You can calculate the magnification. So we look at the size of the image, divide it by the size of the object. Or simply you can write abbreviations that M is equivalent to I, where I is the image size and A is the actual size of the object. Actual size of the object. And we can summarize that in a triangle. I always tell my students, please always have this triangle in mind. Before you carry out any calculation, always first draw the triangle so that you are sure of what you're doing. And I use the acronym I am. I am. So this can help me get the formulae of whichever calculation I'm asked of. Like an example, if I'm looking for magnification, I will remove the magnification, I cancel it out. Then you can see that I remain with image over actual. So that means that magnification is equivalent to image over actual. So that means that is the formula you will use to get magnification. If I'm looking for maybe actual size, I cancel out the actual size. Then you can see if I cancel out actual size, I remain with image over magnification. That means the formula for getting actual size is image over magnification. As long as you have the image size and the magnification, you can get actual size. If I'm looking for image size, I'll simply cancel out image size. If I cancel out image size, you can see I'm remaining with A times M. So it will be actual times magnification. That is going to be very key in our next section that we are going to look at. So let us also remember the units we were using. We were using millimeters. Let us not forget that 1 centimeter contains 10 millimeters. At the same time, 1 millimeter contains 1,000 micrometers. At the same time, 1 micrometer contains 1,000 nanometers. Then you would want to maybe just change directly from centimeters to micrometers. So 1 centimeter contains 10,000 micrometers. So note these down. We are going to use them as we make or we carry out these calculations. So let's look at the first example of where these calculations are going to be needed. So we are going to look at calculating magnification of a photograph or an image. You'll be given photographs in your papers. And assuming I have such a photograph, this photograph was taken from a microscope. And a photograph taken from a microscope is called a micrograph. Micrograph. You will find such terms being used. Micrograph. This is the spelling of the word micrograph. So a micrograph is simply a photograph from a microscope. So the micrographs can vary. It could be a light micrograph, which means a photograph from a light microscope. Could be an electron micrograph, which means a photograph from an electron microscope. So this is a light micrograph. And it's gotten from plant cells. It was gotten from a plant. I think you can see the plant cells. This plant cell has different structures in it. The cell has been labeled P. Now you could be asked a question. They're telling us that Cell labeled P has a real length of 80 micrometers. Real length of 80 micrometers. So that means I'm already having the real size. I'm already having the real size, so I can make a summary before I attempt the question. I have a real size. Real size, remember, is the same as actual size. So I have an actual size of 80 micrometers. Now they are telling me calculate the magnification of the plant cell labeled P. You have several plant cells. If they specify for you the plant cell, please make sure that your eyes only concentrate on that plant cell. So this is our plant cell we are going to consider. Plant cell labeled P. You can see this is the plant cell labeled P. They want me to get the magnification. I remember very well that from my formula of I am to get the magnification, I'm going to use image size over actual. So M is equivalent to image size over actual. In this case, I have my actual size, which is 80 micrometers. But I do not have the image size. So what am I going to do? I'm going to go to this diagram or to this photograph, and I get a ruler. So use a ruler. Your ruler could be in centimeters or millimeters, whichever one you use. But most of the time, my students have rulers that are in centimeters. So get a ruler and measure this cell labeled P. So you have to know where the cell passes in terms of demarcation, that this cell runs from here. The whole of this is the cell labeled P. And if I'm measuring, someone can measure from here to here. Remember, whenever you get your ruler and you place it and you're using it to measure, you're getting image, because this is an image. Someone can measure maybe from here to here. They haven't specified where you have to take the measurement from. So the moment they do not specify where to get your measurement from, just make sure that you measure the longest size or the longest length from that photograph, the longest length. Like in this case, from this cell, I can see that the longest length is from this section. Okay, my ruler ain't straight. Sorry about that. So it's going to be from approximately from here to there. You make sure that you place your ruler well, but it could be from here. This is the longest region, according to what I'm seeing, according to this cell. So I'm going to place my ruler here up to there, and I measure that. That length, that is the length I'm going to measure. So assuming you measure your length and you find out that the cell is 6 centimeters. So that means that your image is going to be, so I don't know what the image is. So assuming you measure and your image is 6 centimeters. That means you can go ahead and carry out your calculation. But according to this calculation, the actual size is in micrometers. So that means that the first thing I'm going to do is to ensure that my image size is also in the same units as the actual size. So in that case, we are going to have our centimeters turned to micrometers, and we saw that 1 centimeter contains 10,000 micrometers. So how about the 6 centimeters that I just calculated? Carry out the cross multiplication. So it's going to be 6 times 10,000, which is the same as 60,000 micrometers. So we are going to have our magnification equivalent to 60,000 divided by 80. So do the math with your calculator. Do the math with your calculator. What answer do we get? Let me also get my calculator. So I'm having 60,000 divided by 60,000 divided by 80. So my answer is 750. So those are the units for, sorry, that is the magnification, but magnification has units. And always the units for magnification is simply a multiplication sign in front of the value that you have obtained. Don't always forget to write your units. So that means that this is the magnification. This is the magnification. That means that this cell labeled P was magnified 750 times. Someone could be wondering where I'm getting these diagrams from. These diagrams, I get them from the course book, the AS course book for the Cambridge AS syllabus. Just a recap, measure the length of the cell in millimeters using a ruler. And if you measure it, even if you go to your textbook and you use the same method, if you measure it, you'll find that it's around approximately 6 centimeters. When you turn centimeters to millimeters, you multiply by 10, that's how you get the 60 millimeters. Convert the millimeters to micrometers, we multiply by 1,000 so that we get 60,000 micrometers. And then we can use the equation to calculate the magnification. So magnification is image size divided by actual size, and that is how you get 60 divided by 80, and you get 750. So that is how we calculate magnification if you're given a photograph, and then you're also given a real size of the object. Let's look at another example. We have a question here. The diagram shows a high-powered drawing of a cell. The actual length of the cell between X and Y is 160 micrometers. Now, in this case, you can see that they have already shown you where the measurement has to be made from line X to Y. So that means I have my actual size as 160 micrometers. But they are asking me to calculate the magnification. What is the magnification? Remember that magnification is equivalent to image over actual. So I have my actual size, but I don't know the image. My actual is 160 micrometers. So what is the image size? Get a ruler, place it along this line. Place it along this line. It should be straight. Place it along that line. So you measure from this point where the cell ends. Not up to here. No. Just where the cell ends up to this point where the cell ends. Now, please make sure that you are as precise as possible, as accurate as possible, because most of the time when you look at the values you get, you're given a plus or minus 1. An example, if someone measures this line, and the line maybe is 8 centimeters. Another person could have maybe 7.9 centimeters. So they will be given a mark. Another person could have 8.1 centimeters. So it's a plus or minus, 0.1 or a plus or minus 1, depending on the kind of measurement that you're taking. So make sure that you're always within the range. So be as accurate as possible. Be as accurate as possible. So we have, assuming we have our length as 8 centimeters, just get the centimeters, turn them to micrometers. We already saw that one centimeter is equivalent to 10,000 micrometers. So still do the math, 8 centimeters, cross multiply. So it's going to be 8 times 10,000, which is the same as 80,000 micrometers. So that means I can bring that into our formula. So I'll have 80,000 micrometers divided by 160. So you do the math, 80,000 divided by 160,000. And the answer is 500. Remember, it's magnification. Don't forget the multiplication sign. So that is our answer. That is the magnification of this cell. You can try out more questions. Remember, the more questions you try out, the better. The more questions you try out, the better. If you need a document, I have a document with compiled questions about this particular section we are covering so that you can practice more. You can comment in the, you can send a comment, sorry. You can send a comment and then I send you that document. Send a comment with your email, then I can be able to share that document with you so that you can carry out as much calculations as possible. I'll send you the document and the answers such that in case you make a mistake, or you can self-mark. Okay, so let's look at another scenario where we are needed to calculate. In this case, we are going to look at calculating the real size of an object from its magnification. So we are having a micrograph still. So this is an electron micrograph. We are going to be given real size, but then they need us to get, so we are going to be given magnification, but then we shall be told to calculate the real size. So here we go. Figure 1.27. This figure shows parts of 3 plant cells. You can see this is one plant cell, another one, and another one. One of the chloroplasts is labeled chloroplast in the figure. So in this case, that means I'll look at the cell, look for that chloroplast labeled chloroplast. This means that you should be able to identify organelles, different organelles inside a cell. As a student, you should be able to identify those organelles. If they mention a chloroplast, can I be able to tell that, oh, this is the chloroplast? They mention a mitochondrion, can I be able to tell this is a mitochondrion? Because if you can't, then you can't carry out the calculation. So they are telling us this image was magnified 5,600 times. So that means this is the magnification. But they want us to calculate the actual length, A, of the chloroplast, labeled chloroplast. That simply means look for that chloroplast. This is the chloroplast, which has been labeled chloroplast. We have other chloroplasts, but they have not been labeled. That means I will not consider them. It's always very important that you follow instructions. So I'm going to get my ruler and place it along this chloroplast. Remember, someone can measure like this or like this, but in this case, I'm going to measure the longest length of that chloroplast. So I will not consider that. That means I see the chloroplast starts from around here up to here. So that means I'm going to place my ruler like this. So place your ruler along the length of this chloroplast following this direction so that you get the longest length, since they haven't dictated where you have to measure from. So assuming you measure that length of the chloroplast, and the chloroplast is 4 centimeters. 4 centimeters, don't forget, turn centimeters to, you can turn centimeters first to millimeters or turn centimeters direct to micrometers. So the choice is yours. The choice is yours. You can do whatever you want. So assuming we are having 4 centimeters. So the chloroplast is 4 centimeters. I know that 1 centimeter is equivalent to 10 millimeters. How about the 4 centimeters which I have measured? So that is how I cross multiply, and I have 4 times 10 millimeters. That means that chloroplast is 40 millimeters. But remember that whenever you're carrying out these calculations, the most appropriate units that are preferred are micrometers. So go ahead and turn this to micrometers. So I know that 1 millimeter is equivalent to 40. 1 millimeter is equivalent to 1,000 micrometers. How about the 40 millimeters that I've measured? The 40 millimeters. Just do the math. So that is going to be 40 times 1,000. That means the size of that chloroplast is 40,000 micrometers. Let's use our formula of I am. Let's use our formula of I am. I am. We are looking for actual. We have image, which we have measured. We have magnification. So actual is equivalent to I over M, which is the same as the 40,000 we have obtained divided by 5,600. Remember, you're dividing micrometers by magnification. So our answer, you get your calculator very fast. What is the answer we are getting? 40,000 divided by 5,600. So if you look at the answer, it's 7.142. There are other values, 857, and so on. But micrometers. Now, when you get such an answer, someone could be like, so how many decimal places am I supposed to use? However, most of the time in the paper, they will specify for you that maybe write your answer to the nearest whole number. Write your answer to 2 decimal places. Write your answer to 1 decimal place. Like in this case, let's just write our answer to 2 decimal places. So that means the size of the cell is going to be 7.14 micrometers. As simple as that. 7.14 micrometers. So that is the actual size. That is the actual size. So in simple terms, measure the observed length of the image chloroplast using a ruler. We saw that the maximum length was 40 millimeters. We changed millimeters to micrometers so that we got 40,000 micrometers. And we use our equation to calculate actual length. So actual length is equivalent to image over magnification. So that's how we have 40,000 divided by 5,600. So for them, they wrote the answer to 1 decimal place. But if they haven't specified, you can write to maybe like 3 decimal places or whatever you want, maybe 2 decimal places or 1 if they haven't specified, but most of the time in the paper, they'll be specifying for you. Okay, let's look at another example where you can be given the same scenario. We have magnification here. Magnification is 5 times 10 power 3. Remember, I told you sometimes the values can be written in standard form. So you should be able to know, interpret what these values are. 5 times 10 power 3 is the same as 5,000. Now, they are asking me, what is the actual size of a nucleolus? So I'm going to look at this image, identify where the nucleolus is. A nucleolus is part of a nucleus, is found inside a nucleus. And from what I see, this is the nucleus and this is the nucleolus, meaning I am only going to consider this nucleolus only. So I'll get my ruler and measure the longest length of the nucleolus. So in this case, place the ruler in different points so that you find out which one is the longest. You can place it like this, place it like this, so that you find out which one is the longest. Because it's not easy to tell the longest or maximum length according to that diagram. But assuming I'm measuring from here to here, and I'm having my image. Remember, whenever you get a ruler and you place it and you get that measurement, that is image. Don't forget that. That is always image. So my image size, assuming my image size is maybe 1 centimeter. My image size is 1 centimeter. So I have magnification, which is 5,000. 5 times 10 power 3 is 5,000. Change centimeters to micrometers. Remember, I told you the most appropriate unit to use are micrometers. So change this to micrometers. And we already saw that 1 centimeter is equivalent to 10,000 micrometers. So they're asking for actual size. I know that actual size is equivalent to I over M. I have my image, which is 10,000. Now divide your answer by magnification, which is 5,000. Remember, these units are in micrometers. So that means that my answer is going to be, you do the math, this goes with this. If you do the math, you're going to find that the answer is going to be 2 micrometers. So this is our actual size. That is the actual size of the nucleolus. So let's look at another scenario. If you have any question, feel free to comment. I'll respond to those questions. Calculating magnification from a scale bar. So what is a scale bar? Now let's look at this diagram. Assuming you're given a figure that shows a lymphocyte. This is a lymphocyte. A lymphocyte is a cell that is part of the immune system, cells that defend us against diseases. So this lymphocyte has been drawn. You have its image. But in that case, they have drawn for you this line. Whenever you see this line is called a scale bar. You're expected to know how to use a scale bar to calculate magnification. You can see the title is Calculating Magnification from a Scale Bar. So what are you going to do? Now, this scale bar always has a figure that is written close to it. And that figure written close to the scale bar is always actual size. Actual. Always, that is actual size. This scale bar itself, whenever you measure it, that is always image. Always image size. Always its image size. So can't I get the magnification I remember magnification. Remember, we use our acronym here of I am. Magnification is equivalent to image over actual. So I already have the actual size, which is 6 micrometers. For me to get the image size, I'm simply going to get a ruler and place it along this line, and I measure this line from this point to this point. So get your ruler, measure that line. Assuming you measure that line, and maybe the line is 3.6 centimeters. That means that the image is 3.6 centimeters. Remember, this value written close is simply the actual size. So change the 3.6 centimeters to micrometers, because we are using micrometers, which will be 3.6 times 10,000. And you're going to have 36,000 micrometers. So get that, divide by the actual size. Easy peasy. And our answer, I think, is going to be 6,000. So our magnification is times 6,000. Our answer is 6,000. So that is how we calculate magnification using a scale bar. So to calculate magnification using a scale bar, just one more time, remember, all you have to do is measure the length of the scale bar. Don't forget the value written against the scale bar is always the actual size. Whenever you place your ruler and you're measuring, you're getting image size. So measure the scale bar. Our scale bar was 36 millimeters, which is 3.6 centimeters, 36 millimeters. 10 millimeters to micrometers. We saw we could get 36,000 micrometers. Then use the equation. Magnification is equivalent to image size divided by actual. 36,000 divided by 6, you get 6,000. Don't forget the units, it's times 6,000. So that is the magnification. Let's look at an example. I always want to show my students as many examples as possible so that they get familiar with the way to do these things. That's why always my students excel. Yes, they excel. A student was asked to use the scale bar shown to calculate the magnification of a cell on a photomicrograph. So they are showing us here. This is the scale bar the student was using. Remember, the scale bar is always the image. The value written close is always the magnification. Now, this is a multiple choice question. They're asking us which method could the student use to calculate the magnification of the cell. We already know that magnification is equivalent to image over actual. We have the actual already, which is 2 micrometers. Can't we get the image? So it means we shall measure this line. Now let's look through the options given to us. Option A, divide the diameter of the cell by the length of the scale bar, both measured in the same units of length. Not really. Divide the diameter. We do not have the diameter of the cell here. Okay, measure the diameter of the cell in millimeters. We have no cell here. They are telling us about the diameter of the cell. No, so that is also not an option. We are supposed to deal with the scale bar, to use the scale bar. So where I see measure the length of the scale bar, it means that is a possible answer. Measure the length of the scale bar in millimeters. Yes. Remember, even if you're using a ruler that is in centimeters, you can turn to millimeters. Measure the length of the scale bar in millimeters, okay. Convert to micrometers, okay. Divide by 2. I see somewhere I'm dividing by 2. What will I be dividing by 2? The image size. Which image size I'll be getting by measuring the length of the scale bar? Turn to millimeters, turn to micrometers, divide by 2. That means the most appropriate answer is C. How about D? D has almost the same flow. The length of the scale bar in millimeters convert to micrometers, yes, but they're saying multiply by 2, yet we are supposed to divide. So no, D is wrong. So the most appropriate answer is C. All right, so those are the different calculations that you're expected to know as a student as far as calculating magnification is concerned. As far as calculating magnification is concerned, we can have a small break. If you're tired, move around a bit, go take some water. Let me also have a water break. The next section we are going to look at is how to use an eyepiece reticule. Going to look at how to use an eyepiece reticule. This is another question, but you can try it out. I'm not going to do it. You can try it out on your own. You can take a screenshot and you try it out. Yeah, so that is your homework. That is your homework. That is your homework. They have given you a cell from a leaf. You have the line XY. They're telling you the drawing is 2000 times larger. So this means this is magnification. And to calculate the actual size of the cell, all you have to get is the image, which is from this section to this section. So measure that line and then get the actual size. Because here they're telling you, describe the steps you would follow to determine the actual size of the cell in micrometers at XY. If you have been following, I'm sure you can be able to attempt that question. These are other questions. You can also still take a screenshot of that and you try it out. You will try it out. Calculate the actual diameter of the lumen of the bronchus using the line PQ. Write down the formula used to make your calculation. Show your working and give your answer to the nearest micrometer. Yes, you can still try that out. To make your work easy, I'm going to upload a PDF with these different questions so that you're able to print them and try them out. So I'll upload that PDF. You'll be able to get it in the description box. And then you can try out those questions instead of, because you can only try out these questions if you have a hard copy. If you're only using a soft copy, it's kind of difficult. Okay, so the last concept about calculations is how to use an eyepiece reticule. So in our next lesson, we shall start with how to use an eyepiece reticule. So see you then. I've just decided to cut the lesson short. So see you then.